or a global patient. Yeah, let's say that. And just I'll put this here. This was the one and only successful email that I ever sent, just the screenshot. So of those 300 applications I made, at the end of the day, one came 3 years later. So this is also just one thing I want to say, and I'll speak hopefully on behalf of everyone on this call to say that if everyone is of African descent, that we are extremely resilient, and that giving up is never really an option when you come from this continent. So if you power through, it will yield good results. Obviously, just assessing your options, and obviously time might be limiting factor in other people's circumstances. But in this case, it was just one thing that paid off. So I think this is a perfect segue into what my research project is, and I'll just give you guys a brief overview. I'll refrain from showing data, as we're working on some manuscripts, and I'm sure you can read the data another time, but also just for the purpose of time and for the purpose of this talk, I don't want to go too much into the data and stuff. So the title of this project is A Global Approach to Advancing Prediction and Understanding of Placental Insufficiency, and the bottom right are just my affiliations, and my supervisor, Professor Katuulino, I mentioned before, Professor Tong, Professor Deschant, and Dr. Non. So this is just a brief outline of my project or our project, and I will be spending the first year of this PhD in Melbourne, Australia, the second year of my PhD in Berlin, Germany, and then back to Melbourne for the third year of my PhD to finish off some things, hopefully. But we all know how PhDs are. This is obviously just the prospective outline, because there might be a fourth year, and I don't know if there'll be a fifth year, but crossing fingers that there isn't a fifth year, because being far away from home isn't the greatest at all times. So for the first year of my PhD, I'll just give you a brief overview of what the project is about and what we aim to do during this first year. So the focus of this project is placental insufficiency. So placental development is crucial for the success and maintenance of a healthy pregnancy, and during placentation, trophoblast cells undergo orchestrated differentiation and they invade the maternal tissue. And this process is key to the development of placental vasculature, which is a network that enables the exchange of nutrients and gases between the mother and the fetus. And in-play placentation may lead to placental insufficiency and related pregnancy complications. And placental insufficiency is a condition defined by the failure of placental vascular remodeling, which leads to placentation, poor placentation, and subsequent inflammation, acidosis, and hypoxia, and associated with diseases such as preeclampsia and fetal growth restriction, or FGR. And preeclampsia is a complex disease affecting multiple organ systems that some of you may know about, and close to 4 million women are diagnosed with preeclampsia each year, with more than 70,000 women and 500,000 babies dying from this disease annually. And it must be stressed that the incidence of preeclampsia is 10 to 30% higher in low middle income countries, such as my home country of South Africa, compared to high income countries, much like Melbourne or Germany, where I'm currently doing some work. And fetal growth restriction, FGR, on the other hand, occurs when a baby fails to reach its genetically predetermined growth potential. And FGR affects 10 to 15% of all pregnancies globally. However, again, the incidence is 6 times higher in low middle income countries when compared to high income countries. And both are key contributors to global maternal and perinatal morbidity and mortality. And the molecular pathophysiology of preeclampsia and FGR is poorly understood. And along with our poor understanding of disease pathophysiology, a reliable and robust predictive test for both preeclampsia and FGR is still lacking. So as we know, placental dysfunction connects preeclampsia and FGR despite their distinct clinical phenotypes. And in preeclampsia particularly, the placenta releases a variety of molecules into circulation, and this further exacerbates endothelial dysfunction and or maternal organ dysfunction. And thus, this makes the placenta a key focus when considering biomarker production, pathophysiology of these prediction, and potential therapeutic targeting. And our team, both in South Africa, in Melbourne, and in Berlin, have taken a pragmatic strategy to concentrate on placenta molecules regarding preeclampsia and FGR, or molecules that may be produced by maternal organs impacted by the disease, in this case, preeclampsia. And these molecules may offer both novel biomarkers, but also may help identify disease drivers and or therapeutic targets. So I like to always put this slide in, but this is just a little, I nearly said a joke. This is just a little old photo from me and my supervisor. We had a little fishing trip And we like to use this analogy that when you're actually looking for biomarkers, especially biomarkers in the placenta, it's like a fishing trip because there's some hits and there's some misses. And you hope at the end of the day that we can get more hits than we can misses. And today, there are some potential biomarkers that have been identified for the prediction and diagnosis of preeclampsia, which include the big ones, soluble FLT, placental growth factor, and biomarkers of placental insufficiency have been identified also in our group, which include but are not limited to SPINT1, SPINT2, DAPK1, and PEG10, amongst many others. But since then, our team have identified 2 potential biomarkers on our fishing trip: thrombosponin 1, THBS1, or cystatin EM, CST6, which will be investigated further and characterized throughout this PhD or for my project. So the first chapter or the first goal was to characterize thrombosponin 1 in preeclampsia. And as I said, what is thrombosponin 1? Just to give you a brief overview. So thrombosponin 1 is an adhesive, an antiogenic glycoprotein with prothrombotic activity. And THBS1 interacts with cell membrane receptors and other extracellular matrix protein and is involved in physiological processes such as inflammation and angiogenesis. And this protein has been extensively studied in cancer and cardiovascular disease, but there are a few articles about its role in preeclampsia. And THBS1 has been shown to inhibit the CAMP-PK signaling pathway in placental trophoblast cells through its membrane encephalous receptor CD36. And we thought that thrombosponin 1 may therefore contribute to the development of preeclampsia by affecting several pathways: the RAS pathway, the VEGF2 pathway, the FGF2 pathway, amongst many others. So therefore, THBS1, along with its receptor CD36 or CD47, may be considered as potential therapeutic targets for metabolic diseases or diseases of pregnancy, such in this case preeclampsia. And its role in pregnancy and placental biology has not really been established, as I said before, until this article that was published before I actually left Melbourne. And it's an amazing paper. They really did some really thorough, brilliant work. And so we decided we should extend on this. And so for this goal, further characterize thrombosponin 1 and its expression in preeclampsia and assess its biomarker potential. So as this is just a brief overview of the aims and the objectives for this part of the PhD. And as I said before, for the purpose of time, I won't be going through any data. So as we saw in this project, thrombosponin 1 was dysregulated in early onset preeclampsia, as well as in the circulation of women with early onset preeclampsia, as well as those preceding diagnosis of preeclampsia. These are women at 36 weeks gestation. And this was contrasting to what we saw in the placenta, where thrombosponin 1 was significantly reduced, whereas it's increased in circulation. We were able to use a cell differentiation model of placental stem cells to identify that thrombosponin 1 is highly expressed or highest expressed in cytotrophoblast cells, and we confirmed this using spatial transcriptomic data. And thrombosponin 1 is increased in HTSCs in primary trophoblast cells exposed to inflammation and hypoxia, as preeclampsia is associated with inflammation and placental hypoxia. And in placental explants, we see that thrombosponin 1 is significantly downregulated by hypoxic conditions. And as preeclampsia is also associated with endothelial dysfunction, thrombosponin 1 was shown to have not be affected by endothelial dysfunction. And to further investigate, we used recombinant thrombosponin 1 and assessed its effect on endothelial dysfunction. And again, we saw no significant changes, indicating that this may not serve as a source for that circulating thrombosponin 1. But in short and in summary, we were able to characterize this molecule and confirm its dysregulation in preeclampsia, and therefore highlight its potential to be seen as a novel biomarker alongside many others, in combination with many others, for preeclampsia and diagnosis. And then just quickly moving on to the second part or the second chapter, which was also during my time in Melbourne, was to assess CST6 or cystatin EM and legumain, LGMN, in preeclampsia using the same approach. And just a brief introduction of what these molecules are. So CST6 is a member of the cystatin family of protease inhibitors, and it's been implicated in a variety of biological processes, including tumor suppression and promotion, but its role in pregnancy is not really understood, and in preeclampsia. And cystatins play a crucial role in regulating endometrial development and placental remodeling, processes essential for establishing and maintaining pregnancy. And this has been shown in various species such as sheep, cows, pigs. And CST6 demonstrates a high affinity for cysteine protease legumain, and legumain is found within lysosomes, and it is involved in various cellular function, including antigen processing, neuronal cell death, and tumor cell invasion. And we see that CST6 regulates legume function to inhibit proteolysis, and has been shown to support implantation and placentation in the bovine uterus and as well as pigs. So therefore, we also aim to do the same and characterize CST6 and legume in preeclampsia. Again, the aims are very similar as we use the same well-established biomarker validation pipeline. And we found that CST6 was dysregulated in the placenta of women with early onset preeclampsia being increased while legume was decreased. And the same was observed in women preceding diagnosis of preeclampsia, and this was in circulation. We again confirmed the placental cell type expressing this, expressing CST6 in legume, and we validated this using spatial transcriptomic analysis of differentiated organoid culture, publicly available data set. And again, we exposed syncytial trophoblasts to inflammatory cytokines, and we found that CST6 is upregulated, exposed to hypoxia, whilst there was no change with inflammation for both legume and CST6. And endothelial dysfunction had no effect on legume, but leads to a reduction of CST6 expression. And again, looking at the effects of recombinant CST6 given its dysregulation in preeclampsia, we found it had no effect on endothelial dysfunction and legume expression, and does not affect tube formation or angiogenesis. So that was just, sorry if I went over there, that was just a brief outline of what I did in my first year, and now I'm currently in my second year in Berlin, Germany, and we using this to, the work I'll be doing will be comprising chapters 3 and 4 of my PhD, and these include identification and characterization of novel pathways and molecules differentially expressed within placental insufficiency using bulk RNA sequencing and single cell or single nuclear RNA sequencing. So just a brief overview. We don't have the data. Everything's been sent for sequencing this week and the week before. So currently the data analysis is underway. And Michael, I'm sure maybe you could help me out actually, as I'm currently being trained on all the bioinformatics and all that kind of analysis. But just a brief intro. Multiomics is a cutting-edge technique that enables the simultaneous analysis of several molecular components and changes with high precision. And through combination of different targeted and untargeted omics methods, novel and key pathways may be discovered. And this broad approach offers a bigger net in our search for molecules important in the pathology of placental insufficiency. And the integration of omics data with clinical information will allow us to therefore identify molecular pathways key to placental insufficiency. And previously, this paper at the bottom was a paper we published at the end of last year, a review. And the paper at the top, I should actually change because that is currently has been published in hypertension. Previously, our team has used spatial temporal multiomics profiling combined with single nuclear RNA sequencing to investigate the early stages of early onset preeclampsia and to identify candidate biomarkers and pathways. And this is kind of the goal going forward as for my time in Berlin. So by understanding the cellular pathophysiological mechanisms underlying placental insufficiency, we aim to improve the understanding of specific phenotypes of disease, and therefore we may develop new ways of predicting pregnancy complications such as preeclampsia and FGR. And interestingly, there's only been like a few limited large-scale omics analysis of the placenta, and one study provided a complete analysis of the human placental transcriptome using single cell from patients with preeclampsia at term, late onset preeclampsia. And importantly, it was very important for the study, that they utilize a well-characterized patient cohort, which has been a limitation of previous studies involving this kind of technology. And so therefore, we aim to identify novel candidate biomarkers and pathways from placental samples from Australia. So the bottom is just kind of the recruitment criteria for the patients that we use, and the sequencing is currently underway. Oh, and there we go. So this is kind of we just aim to use these techniques to characterize and validate novel candidate biomarkers for the potential use as a clinical diagnostic aid for placental insufficiency. So through combination of these targeted and untargeted omic methods, we want to identify crucial pathways and identify novel biomarkers for these pregnancy-related complications. And what we aim to do in combination with what I previously discussed was we want to evaluate placentas obtained from low middle income countries using, again, bulk RNA sequencing or single cell RNA sequencing. So bringing it way back to the beginning, this is very close to home because obviously this has potential impacts for South Africa and hopefully for Africa as a whole. And as we know, the degree, sorry, that image shouldn't have come up, but the degree of severity of placental insufficiency does differ between low and middle income countries and high income countries. And if and why these differences occur in the severity and prevalence of symptoms, as well as the difference in clinical manifestation of placental insufficiency, is still the subject of ongoing research. So again, bringing back Lena, Kathy, and Clive from South Africa, who have the biobank and also have labs there. We hope to recruit patients from Tygerberg Hospital and send all those samples up here to Berlin, where I'll be based, to sequence them, and I'll be doing the analysis. And hopefully we can compare placental samples from low and middle income countries with placental samples from high income countries, and see if any we'll assess global gene changes in pregnancy samples from low and middle income countries, and compare that to global gene changes in placenta from high income countries, and see what differences there may be. And taking these findings, I'll return to Melbourne and then validate these in a few of the international cohorts that are present there in Melbourne. And hopefully we can come out with some groundbreaking findings, as is the hope of every PhD student. And as I end off on my presentation, and I just want to also thank you all for your time today. And sorry if I went a bit long, Wealth and Michael, but just the importance of friendships and the support structures that you have. These are some of the lovely people that I was able to meet and stay with during my time in Melbourne. I learned an immense amount from them. It's all different brains, all different personalities, a lot of different different nationalities, different cultures that you have to get used to. But essentially combining the strengths of each different culture, each different country is essential to scientific endeavor, essential to scientific success. And yeah, I just think it's important I cannot stress enough the importance of network and collaboration, aside from just being, and not saying this of myself, but if you're brilliant, that's great. If you're talented, that's great. But it's the people that you surround yourself with, your support structure, that will essentially determine how successful you can be within whatever endeavor you decide to pursue. And this is just a quick little summary of my time in Berlin. I haven't had much time. I've only been here for a month, but learning a lot. Again, I ran a race when I was here, nearly lost a lung, but an amazing time so far. And then another shameless plug to again stress the importance of pursuing your passion. During this PhD, I released a single, which you can again use the QR code, a shameless plug, sorry about that. But wanting again to stress that we have time to pursue our passions and our hobbies, and it's also important to pursue your passions and your hobby to keep yourself sane during these endeavors. And those of you on the call who have your PhDs already, you might be well along in your career. I'm sure you can maybe back me up on this one. So in summary, we conduct research for a living. We're good at it. There's so much information available out there. We're good at compartmentalizing this information. So take whatever information is, do your reading, understand your environment, whatever goal you're working towards, there's so much available out there to allow you to cater your kind of application to how you can go in and achieve that goal. It's not easy, but build your network. Some of us aren't as confident as others. Some might be introverted. Some might be too extroverted. But this, I think, is a good skill that we should learn, and a good skill that we should actually nurture and take time to build, because it's super important for our careers going forward. And again, I say this, being a proud African, don't underestimate the power of kindness. And throughout my time working in South Africa and with colleagues across Africa, we are incredibly kind people. And I think that goes a long way in the broader scheme of things, especially coming overseas, mixing with different cultures. And I'm sure some of you who are overseas or have gone overseas to study can definitely relate to how difficult it can be to integrate, especially if there's a language barrier. Define and keep redefining your goals. I think it's always important to go back to the drawing board and reassess your position, understand where you are and what you need to do, what you need to change, because adaptability is also key in this career, in this field. I'm just speaking now specifically from a science perspective. And something I've realized, our biggest obstacles indeed are ourselves, and that's our only limiting factor, because the world is our oyster and we can thrive regardless, as long as we make that switch in our mind. And in doing that, I think it's just important to ask for help. And my partner always tells me, and she's always right in this regard, that we should never be the smartest person in the room, and that is true for everything. and never be shy. We are not all smart enough to put your pride aside, sorry, in that we should not be able to ask for help. I think it's super important in order to get forward in life. Understand our environment. That's something I also put on here because coming from South Africa, I speak just as a South African working in the South African environment. We're extremely resource limited. There's limited funding, limited equipment that we can use. So it's important to also know how to act within those means and what we can, and to ensure that we get the best out of these environments. And moving on to the next point, I think it's a good segue, is that this is extremely challenging and it's not going to be easy. And this is something that I also realized with my time in Berlin. The first month here was a culture shock. I didn't speak the language. I still can't really speak the language. It's a difficult language. But again, as long as you remain confident and passionate and kind, I think things will be a lot easier for you going forward. And I say this to the up and coming young scientists that might be on this call. And passions outside of work are essential to keep you sane. And at the end of the day, have fun. And I just put these in here because I thought just to remind us in case we forget, but to be African is to be special. And so the scientists and researchers, clinicians that are listed here are few of so many that are absolutely inspiring, that have come from the African continent and that continue to make, some have passed away that are on the slide, and may they rest in peace, but that they continue to make inroads worldwide and globally, and they impact globally. So we, as Africans, we have capacity, we are brilliant, and nothing should actually hold us back on a global scale. And my cheesy statement to end off the talk, we are Africans and not Afrikaans. And I put a little cheese emoji there just to highlight that, how cheesy it is. So while resources may not always be the most abundant, and our potential for scientific discovery remains limitless. And the world as a whole might see limitations in our surroundings, but let's be clear, those limitations do not define us. The human mind is a boundless frontier, and ours, fueled by the rich heritage of this continent, is no exception. And the world needs the unique perspective and solutions that come from African scientists tackling problems in their own environment. And our ingenuity and resourcefulness are exactly what can help us solve some of humanity's most pressing challenges. And the rest of the scientific community needs us at the table, not just for our discoveries, but also for the fresh lens we bring to scientific inquiry. And through collaboration and by sharing our knowledge and expertise, we can achieve breakthroughs that would be impossible for any of us alone. And we are resourceful, and we are fueled by curiosity, and we have a passion for progress. And I just implore us to keep pushing boundaries as we proceed. And this is just a bit of acknowledgement to everyone that's been involved in this journey so far. And I want to thank them a lot. And the main acknowledgement will be my family, my friends, and my partner, Caroline, who's actually on her way to Berlin as we speak to join me. Without their support, none of this would be possible. And I just want to stress also the importance of having a good support structure, which might not be the case for a lot of people. So yeah, reach out to colleagues, reach out to friends, care for each other, support each other, advocate for each other. Science is extremely competitive, but it doesn't need to be at the end of the day. And this is just a slide I wanted to put. That's my final slide, just to highlight some international opportunities for the young researchers that might be on this call, or young graduates. A lot of them are very German-based as of now, as I'm still figuring out a lot, but we've got the Rhodes Trust, the DARD scholarships that are very good, GLORA, which is the Global Health Initiative, started up here in Germany, and the Berlin University Alliance, and they have amazing opportunities for all kinds of people from every different nationality. And yeah, great opportunities to go abroad, which is great for expanding your environment and your skill set, and just for learning a different experience, taking yourself out of your comfort zone where you can do the most growth. And I just want to also put a big mention to the Crick Africa Network, or the CAN Fellowships, which is affiliation with the Francis Crick Institute in London. My partner was an ex-fellow, and I currently have a few friends who are now on the fellowship now. This is an amazing initiative for early career researchers and postdocs who are looking to take the next step in their career. And this is, if you don't know about this, some of you might know about this, but I just want to say that this is an amazing, incredible opportunity to take that next step. You get to spend some time at the Crick Institute, do some research, incredible funding comes your way and supports your growth in Africa. A lot of the past fellows have gone on to become professors, start their own labs. I think I can't remember his surname, but Yao from Ghana has started his own biotech company, and he was a previous CAN fellow, and he's doing some amazing work for scientists in Ghana, providing training opportunities, jobs, and building amazing infrastructure there. So yeah, thank you so much to Biospheria for today, and I'm sorry for blabbing so much. Thank you all for this incredible opportunity, and if any of you have any questions, happy to discuss now, but also there are my details, I'll leave them up, and yeah, thank you so much.
Yeah, thank you very much, Stefan. That was a very wonderful presentation. It was inspiring, it was encouraging, it was motivating, and your storytelling is amazing. Taking us through your journey, letting us understand that the challenges do not really define us, and we are very resilient enough to overcome these challenges that come with conducting research, even as Africans that we are. I'm sure a lot of people have a lot of questions to ask, so I think we can allow them to raise up their hand and why they can unmute themselves to ask the question, and also other people can just put questions on the group chat. So while we are waiting for them, I can just ask my own question. So how are you settling down in Berlin so far?
Oh, the good card of I was waiting for the science when I was like, please no science, but there's that. But no, it's been really tough. I'll be honest, for the first month, as I said in the talk, it's it's a completely different language, a completely different culture. I mean this in no way to offend Germans, but they're a bit more reserved than I would say Africans. We're all like, so that's something you have to really get used to. And there's a bit of imposter syndrome that comes, and I'm sure, Michael, you said you had Massachusetts now. I'm so sure there's times where you feel like, why am I here? Why am I doing the right thing with my life, etc. But slowly, it's getting there. It's getting there. Thank you. Yeah, just pushing hard. Yeah, thank you.
Thank you. So yeah, if you have questions, you can unmute yourself and ask or put them on the chat. I can read them out for you when they have them. So while I wait for people to also I can ask my question. So what tissue sample or what biological sample do you collect for your research, for the placenta?
Awesome. That's a perfect question. So back in Melbourne, we work with a placental cell line, but we also are very fortunate that we actually operate just above the clinic. So placenta comes from either selective cesarean, and we are able to then sample from the placenta directly. So we take placental explants, we isolate primary trophoblast cell from term placenta, and now for the sequencing, we embed placental tissue in FFP blocks, so from PFA, paraformaldehyde embedded, and then we have RNA later, we call them Tic Tacs. Quite a funny thing. We just take small bits of placental tissue, store it in RNA later, and we send that for sequencing along with the PFA blocks or FFP blocks. That gets the single nuclear spatial transcriptomic stuff. Yeah.
Yeah, very cool. Yeah, thank you. So we have Joseph. So Joseph, you can unmute yourself and ask your question. Yeah.
Hi, Joseph.
Hi, good afternoon. That was a very interesting talk. Thank you so much. So I just had two quick questions.
Perfect.
The first is, your project is really fascinating. And then I was wondering, is this study exclusively in humans, or you have cross validation in rodent models too? Or are you just working with humans?
Oh, that's perfect. So this great question. This project at the moment is quite a discovery-based project. So we're hoping to find any molecules potentially dysregulated in preeclampsia versus normal or fetal growth restriction versus normal. And then we take that to validate that in vitro and in vivo models. We do have mouse rodent models, at the labs. But as of now, we mainly just focus on actually identifying molecules that are dysregulated and then publishing those as hits. And then we'll try and validate that further in models and see if they can have utility as a biomarker. Yeah. So we will take it further, hopefully.
Yeah. That's interesting. The reason why I asked is because I'm actually working on a similar project right now, and then it's also biomarker discovery basically.
Yeah.
And then I found out that most scientists like to do cross validation with humans, with rodents, and things like that. That's nice. The second question is so I'm curious about the IRB process over there in your country, Institutional Review Board. to get approved to work, to collect these samples, right? For example, over here in the US, sometimes it's a pain in the neck, to get approved for studies
Always
like this. You have to go through a lot of process, a lot of training and all that. So how does it look like over there in your place?
So I think this is also, I don't want to call it a benefit because it's not a benefit. So the disease burden is extremely high in South Africa. So I want to say that that's a benefit of having, but we have a lot of samples. And in terms of collecting those samples, it was just a process of me actually, for the first part of my PhD, I actually had to write a massive ethics application to the Human Research Ethics Council at Stellenbosch University, and get a whole bunch of MTAs and DTAs together so that we could ship samples. So much like the paperwork I'm sure you went through, I definitely think it was a bit more relaxed when it comes to shipping samples from South Africa, as it must. I'm sure the US must be quite intense. But yeah, there's an ethics that's been established. We have all the protocols in place for shipping, and yeah, we have an agreement between Melbourne, Germany, and South Africa only that's how we can share those samples. Yeah, we're very lucky in that regard. But it wasn't easy to establish that protocol. It took a whole year. So yeah, to answer that question, it's probably a bit easier than the States, but it's still difficult at the end of the day.
Yeah, I can imagine that. All right. Thank you.
That was a precious sample. Yeah. Very precious samples. Yeah.
All right. Thanks for your response.
Yeah, thank you, Joseph. So we have Shion. Shion can also unmute himself and ask. We also have some questions on the group, on the chat. I'll read that later. Yeah. So Shion, you can ask your question.
Hello, please, can you hear me? Just to confirm.
Yes, we can.
Yeah.
Okay. Yeah. Thank you so much, Stefan. I actually joined in your section late, so probably the question I want to ask. So it's just maybe you might have talked about it in the previous slide. So I kind of find out that we're kind of using bulk and spatial transcriptomics to look into the placenta for the particular research you want to do. But I want to ask the major thing I want to ask there is are you looking at the two technologies side by side? Are you trying to cross-validate stuff, or you are using just a single technology for that?
Oh, great question. It's actually something we discussed this week, actually. So we have limited samples for this, the spatial transcriptomic, and limited samples for the bulk sequencing. So what we're hoping to do, and this is way out of my pay grade because it's something I'm learning, but we have amazing bioinformaticians and data scientists working on this stuff. They're hoping to and this is a term I also learned this week, so forgive me if I misuse it, but use confounder modeling, and they're hoping to almost get away with, because some of the samples are matched. So we have samples that we sent for spatial transcriptomics and samples that we sent for bulk sequencing. And we're hoping in some way we could cheap out and kind of like confounder modeling, actually overlap them in some way or form. Obviously, there's a lot of samples that we send for bulk that we can't do that, but with the spatial stuff, we're hoping we can overlap and use it in that way together and develop like a massive model, or like get some crazy data out of that. But yeah, I'm still learning this a lot, so forgive me if I didn't answer your question properly.
Okay, yeah, thank you so much for that.
Okay, yeah, thank you, Shion. So I think on the group chat, there's a question about what were the challenges in how easy was it to get placenta samples? Any challenges encountered, ethical barriers, and yeah, so that was the major question.
Okay, I see now, especially after Naziruddin's comment on that too. Great question. Yes, definitely a lot of ethical barriers, and that's why we have a whole protocol and assessment by the Human Research Ethics Council that honestly took a year for them to assess our application. So they can either reject it or tell you to modify it in some way or form. But in saying that, once it was approved, we had a whole bunch of patient consent forms and child assent forms that were translated in a variety of languages. Like I'm sure a lot of you are familiar with the whole recruitment process. So we have a team of research midwives that go out into the clinic and they consent patients before they collect the placentas. So, and a patient can also withdraw placenta. In that case, we have to if we've processed it, we have to discard those samples if the patient chooses to withdraw. So yeah, in terms of ethics, it's quite sound. And we consent, it's informed written consent from all patients in their language of choice before they decide to give a placenta. In order to not, obviously there's probably some religious barriers and stuff, but we do our best to navigate those as best we can, and hopefully don't breach any ethics protocols throughout this whole process. But luckily for now, especially from the Melbourne side, I can tell you they've been recruiting for ages, and same in South Africa. Nothing's gone wrong, and yeah, so we hope nothing goes wrong for this project. Hope that clarifies a bit more.
Yeah. Thank you, Stefan. So another question here is, you employed multiomics as part of the approach for your research. So could you briefly mention the specific omics analysis you employed in your research?
So the omics analysis right now is currently underway. So specific techniques in terms of like platform, or what specifically do you mean by?
I think that would be the kind of maybe data being sequenced. I think you're kind of, since you had a multi-omics approach, so you did a lot of transcriptomics, maybe genomics and other stuff.
Okay, so is it about the analysis or more just what we're actually doing with the samples?
I think more of the data generated and the kind of analysis being done. Yeah.
Okay, cool. So Abdullah Fiz, thank you so much. I hope I pronounced your name right there. Thank you so much for the great question. And this is exactly to Sean. This is something completely new. I'm very much a lab scientist in this way. The dry lab stuff is something completely new to me from my knowledge as of now and coming around this whole project. So we've got samples that we sent for spatial transcriptomic sequencing, and then samples that we sent for bulk sequencing through Novogene using various platforms. So this is just getting an overall genetic transcript at a single cell resolution for the spatial transcriptomic, and then a general resolution for bulk sequencing. And from that, we'll employ, I don't do any of the QC data cleanup, but the deconvolution step is something that I'll be learning and taking part of, and that will be done through using RStudio. And then we'll make use of a variety of publicly available pipelines. One that I know that I'm currently, I just say this as I had this meeting on Friday last week about what I need to actually learn. And one of them is I'm going to be using CyberSort X, and firstly trying to validate previous findings from a paper where they employed this package. And then with regards to and that's just as my training, and then I'll apply this to our data set that we get. And with regard to the spatial analysis, I'm doing none of that data analysis. That's actually my PhD twin, I call her, because we're two joint PhD projects on this project. I'm more of the wet lab scientist. She's very much a dry lab scientist, and we work together. And she's analyzing a lot of the spatial transcriptomic data using the Suret package on RStudio, which is quite well established for analyzing these single cell data sets. And she's going through establishing the matrices and then deconvoluting the data and then working with that as it goes forward. And I hope that answers your question, and I apologize, as email me in a year and I'm hoping I can answer that way better. But thank you.
Yeah, he has a follow-up question.
Proteomics. Oh, there we go. So actually, Greg, you're on it, man. I feel like, do you want to swap here with me? Because you actually did so well. We actually plan on doing spatial proteomics on some of the placental samples, and my training for that, we're still having meetings to test the feasibility, just because I'm limited with my time in Germany, because we can only kind of do this kind of state-of-the-art like analysis over here in Berlin at the MDC, and we're currently in discussion to test feasibility of spatial proteomics on placental tissue. And I'm hoping in January next year, we'll be able to use some of the FFP blocks that we use for the single cell RNA sequencing to kind of use that for the spatial proteomics. And that's going through a group at MDC, Dr. Fabian Kosher, or Professor, I think, Fabian Kosher, who's doing some amazing works in spatial proteomics field, especially in cancer research. I do recommend you guys, if you don't know about him, check him out. He's really amazing, doing some good work. But yes, again, learning. I've only been here for a month, so a lot of this is very overwhelming. I'm still learning it all. Thanks.
Yeah, okay. Thank you, Stefan. We have more questions. I'm just going to read this couple together so you can answer them together. How did you cope with the season in which you had to grapple with the multiple rejections? Any useful lessons from them that you'd like to share with us? What are the key implications of the findings that you made about the preeclampsia biomarkers, and what potential recommendations could they lead to, maybe in terms of drug development? And so, yeah.
Perfect. Thank you so much. Thank you, Walter. That's a great question. The first one, quite a deep one. In terms of grappling with those multiple rejections, I'll be honest with the people on this call. I was in a very dark space. It's not easy. Maybe some of you can relate to these kind of downs that you have. You completely feel like you're worthless. You thought going this route, checking boxes, BSc honors, MSc, you thought you were going to be this amazing scientist, and then you hit a wall and you're kind of told that you're not really that good actually, and you can't compete. To be honest, at the end of the day, I had to kind of pull myself up by the scruff of the neck. I had a good support network, and I'm very privileged, I know, to say that I had a good support network, and they helped me really gain some perspective. And I got lucky. I'll be honest, luck does play a certain role in this situation. I'm lucky that I ran into that old professor and I was able to get a job interview and land the job that kind of put me on the path that I'm at now. And any useful lessons from that, I would just say, really be resilient. You have to grow a bit of a tough skin. And it's horrible to say that because it shouldn't be that way, but unfortunately it is. And you kind of just got to use each failure as a lesson to move forward. I kind of like to just say fail forward. That's the best thing you can do in life. I mean, science is all that. A lot of the time your experiments are failing, and then the one time it works is when you try to write up your thesis. So I'm sure we all know that feeling. And then key clinical implications for the findings. So the group now, they actually doing some major work while I'm actually here in Berlin on some other molecules, but we hope that we can improve diagnostics and prediction of preeclampsia, and that we could hopefully any novel biomarkers that we discover and validate in a clinical cohort or in vitro and take to a clinical cohort. We hope that we can combine that, or maybe robust on its own, but we could combine that with existing biomarkers and then develop a more robust and cheaper alternative for what's existing out there, even though there's only really one or two tests out there for predicting adverse pregnancy complications. And yeah, we hope that we could contribute to the field in that we could develop a more robust predictive test for these disorders, especially one that could be practical, easy to use, and cheap. Because my goal at the end of this whole project is to hopefully make an impact, but also an impact in South Africa and hopefully in Africa as a whole. And be it that the training I receive overseas, I can come back to South Africa, start a lab there, and hopefully train young scientists up within this field. And number two, develop a test that we can actually take to clinic, because the burden of disease, as I said, it's 5 to 6 times higher in low and middle income countries. And that isn't due to the fact that we are molecularly different or physiologically different to people in high income countries. It's just a systematic issue in that our women present to clinic later. They might be stuck in a South African context, they could be stuck in a township without transport to the hospital, the clinic. It might not be on their priority list. They might have to feed their family as opposed to go to the clinic, etc., etc. So there's all of those factors that we have to take in consideration. And I'm hoping in some way we could take a point of care test to them and stratify them in that way. Commit them to ICU before symptoms, onset of symptoms, and hopefully save lives, both mother and child. Yeah. Sorry, that was a bit waffly, but hope that answered it.
Yeah, thank you very much, Stefan. So while I wait for maybe a few more questions, I have one question. Did you collect time points, like samples, like across the different pregnancy stages to check for like these different, maybe different genes are probably expressed at different time points of the pregnancy? So did you do that in your research?
That's a brilliant question. So I have colleagues who are doing it at the moment. So we have placenta at different time points and blood at different time points, or plasma that's been collected at different time points. And you're able to measure changes in gene expression, changes in protein secretion across gestation, as you said. Right now, I'm just more focused on the different, so as preeclampsia presents as different subtypes, or placental insufficiency might occur at different stages. We're kind of focusing on early onset, so less than 34 weeks preterm. And then late onset or at term at 36 weeks and greater. So those are kind of the 2 time points now that I'm focusing on. Hopefully, when it comes to validating these in vitro or in vivo, we'll be able to use cross gestation once I get back to Melbourne. Yeah.
Yeah, nice. So yeah, thank you very much for So there's another question on the chat. So what's your view on the need for more African institutions to collaborate with international partners to train graduate students and also to be able to conduct cutting-edge research? How can we address this without creating the problem of helicopter science? So, yeah.
Well, that's a stunning question, and I'm sure a lot on this talk I've had those like those same questions or thoughts as we go on. You're so right. What can be done? It's something I've been thinking about throughout this journey that I've been going on. I've had many discussions with my mentors back in South Africa about how we can avoid this problem of, and forgive my terminology, but like where you just become a sample manufacturer, a sample producer, and you ship samples to the global north. They do all the state-of-the-art stuff and get the Nature papers and all the funding, and we left with just the infrastructure isn't developed as much, or it develops at a slower rate by virtue of lack of that funding, lack of competition, because we don't have those papers, etc., etc. But I do think it's extremely important for African institutions to collaborate with international partners and to have these kind of joint PhD programs in way, or joint training opportunities in way, even if it's semester abroad or something, because that exposes our scientists to other techniques, learning new skills. For example, I would never be able to learn this spatial transcriptomics, all this analysis if I was back in South Africa, just because the infrastructure that we had for this kind of research is just not there yet. So an opportunity like this, and through these collaborations, it's essential for me to learn these skills, and hopefully one day could bring these skills back to South Africa, start my own lab, start training South African scientists, maybe from North Africa, middle of Africa, east, west, you know what I mean, on these techniques locally. So it isn't so taxing, because I think you're so right in having this question, and it's not a concern, but it's something that I think in future will hopefully be sorted. Because also another thing I can say, and this is maybe something that could be a discussion in this group, maybe wealth, I don't know, this could be a topic for future, but it's like something I've run into a lot is visa issues. So as a South African with we call it the green mamba, with that passport, I have to apply for visas every single way I go. International conferences, I have to get a visa. I have to go for medical exams. I need the blood of this person. I need the thing. And I say that as a joke, but it's more like you have to jump through a lot more hurdles than our compatriots in other countries, be it the higher income countries or like Europe, just say, for example. So I'm hoping from that perspective, science will just become easier for Africans in that way. So we're able to, because like I said in the talk, we have brilliant scientists, we have capacity, but we're just limited by things that are out of our control. And hopefully these collaborations will alleviate those problems.
Oh, okay. Thank you very much for your for addressing that. That's very important that we find these collaborations between African institutions and institutions outside the continent. We foster this collaboration because we are very, very intelligent. We are just resource constrained. So it's just
exactly.
So we are all intelligent and smart. So once we get into the space with enough resources, I see us flourishing. So
exactly. And also, like, from that point, also, like, even though they hold all the funding in that, they just have majority of the funding there, it's like nothing also stops African institutions from collaborating with each other. Because I do think that as African institutions, as African scientists, we need to advocate also for each other. We can't just rely on this whole thing of, I will just go to Europe to practice science and to become a better scientist and stuff. It's like we need to build capacity here and start here first. And I was just very lucky that I got this opportunity to go, but we made sure, sorry, for this project, that Africa was included, in this case, South Africa. So I do think that it's international collaborations also exist within a continent, and in Africa we have brilliant institutions, brilliant scientists across the continent, and we should be all working together on African problems also.
Thank you very much. Thank you. So is there any other questions Can raise up your hand, unmute yourself, or drop questions on the group. Yeah. Okay. All right. So I don't know. I just a fun question. Your undergraduate university, is it a music university? Because I know they have like a choir and they do a lot of music stuff. So yeah.
So you said, is there music at the university?
Yeah. Did you do music? Did you study music at the university also with your bachelor's, or is the institution like a musical institution?
Oh, no. So they
Language.
have music, and they've got a very good music school, and the same at UCT. But no, I didn't study. I wish I did, because that's one thing where I don't know too much. But I started high school was in the choir and did all the that's marimba and steel things, and yeah, played, learned how to read music and stuff. But yeah, learned how to play guitar, and then, like I said, it kept me sane, and then met up with some friends during university, and yeah, and then the rest is history. Started the band, and yeah, I was very lucky and very privileged to do that.
Yeah. So you have a question now. So how do you make time to do music in grad school? Sometimes difficult to keep up with all the grad school stuff. So I know your PhD program is kind of fast paced because you're not taking coursework, I believe.
No coursework, no.
Okay. So yeah. So how do you, with all the fast paced research, so how do you keep up with like doing music, and does your lab or program support this? Do they know you do this on the side? Yeah.
Oh yeah, so they do know. I mean, it was so funny. At the one conference, they actually put it up at the end of the one. I was like, why? But why? But yeah, so I guess it's also just prioritizing and making time for the things that are important. Because like work is work. I had many a weeks where I was in the lab 24/7, like I'm sure a lot of you on this call have had, where you actually have time for nothing. But it's super important to me, and therefore I will make time for it. And yeah, like, I mean, I have to do courses and stuff for my German graduation because I have to get credit points and stuff, and you have to attend certain courses and seminars and stuff. But yeah, I guess one thing that I've learned throughout this my time as a PhD student is like time management, prioritization. It was something I wasn't really good at until I started this, and I realized, no, I needed to be good at this because it actually, yeah, you got to make time for these things. And with the music thing, I'll be honest, I should have been better as of late, but well, I've struggled at the moment. So, Michael, I've struggled. It's been a bit of on a back burner. And yeah, I think does your lab or program support this? I mean, yeah, as long as it doesn't cut into your work, and as long as they're getting the data at the end of the day, I don't think anyone would be upset. So yeah, they definitely love the fact that we do other things, and I would encourage everyone to do things outside of work, because you'll stifle your creativity in the lab if you don't, to be honest, and your thinking. You'll burn yourself out.
Yeah, nice. Thank you very much. I think we don't really have any other question for now. So round off soon. So I just have one last question for me. So how do you think, because the major barrier or the major challenges to actually conducting this research ourselves is the resources we need. I think in terms of like brain power and manpower, we have those. So how do you think Africa as a continent, or researchers in Africa, can better put themselves in a position to get grants and get this funding? What do you think they could have? What do you think they're not doing that they can do, or what do you think they can do better? Yeah. So
yeah, that's great question, and it's something I can honestly say I don't know the answer to. But what my opinion is that, and much like Paul's kind of comment, it's like through international collaboration, but we advocate to put ourselves first, if that makes sense. So it's like, we can't always think that the people with the funding and stuff, that we need them to make inroads. We got to also understand that they need us in a lot of the circumstances. We have a interesting, diverse population with a diverse set of problems that are key to like discovering, making major breakthroughs in the global sense of things. So it's like, as Africans, we need to take the lead on a lot of collaborations, and not just let those with the the most publications, the highest H index take the lead in that case. So I do think it's important that we advocate for ourselves and fight for ourselves in those settings. And I think that's a good way forward in terms of building African infrastructure, allowing us to be competitive for grants. I do think it's just unfortunate that a lot of the funding schemes don't really look at Africa first. But I would say, and I think Australia is a good example, they kind of isolated down there at the bottom of the earth. And they've really developed their own funding structures, and really their governments have put a lot into science and to build that. Obviously, in Africa, it's a different kind of conversation, given like the way governments work here, how much money we have. But I do think it's good if you take care of your own and then look elsewhere. And yeah, so it's like prioritize infrastructure first in your country and then look elsewhere for these international collaborations. And then when you do get those collaborations, really advocate for yourself and say, no, I'm an equal partner in this, and not just someone who's, just happy for the paper or happy to get the name on the grant. It's like, no, we should be PI or joint PI or something. It's like, I will have a collab, but then you take one of my students and I'll take one of your students. Like you can make these sort of arrangements to benefit students, benefit early career researchers, benefit professors, yeah. And that's why I put the Crick Africa Network thing, because I think that's an amazing initiative. It's also a collab with LifeArc, which is an amazing, massive pharma biotech company. And they're pumping a lot of money into Africa, into developing African scientists. And I do recommend go to their page and look at the past fellows and look at some of the work they're doing post their fellowships. Yeah. So really cool stuff.
Yeah, thank you very much. I think for me, this session has been inspiring, it has been motivating, it has been encouraging, and just brings back the whole just resonating like all the thought I've had in my mind about always like going back to Nigeria to like do research, and personally for me. So it's good to see that the other African scientists that are also thinking in this light about the challenges that we face in Africa. So before we round up, I think maybe Wealth can say a word. Well, yeah, thank you for coming around, Stefan. I will hand it over to Wealth now.
Thank you, Michael, also for moderating this. You've been an absolute G, and appreciate all your help here, and all the best with your studies, man. I hope it goes super well there.
Thank you. All the best too. Yeah.
Yeah, thank you. Thank you very much, Michael. And thank you, Stefan. Okay, Abdulafiz wants to speak. Maybe you would speak and I'll go ahead. Okay. Abdulafiz.
Yeah, perfect.
Yes, we can.
Oh, that's perfect. That should be perfectly fine.
All right. Thank you, Hafiz. So Stefan, I really don't have much to say. We are grateful that you accepted our invitation and that you were also very willing and happy to share so much about your work and your journey so far. I've been inspired and motivated. I've also learned a lot. And like Michael said, seeing some perspective that I strongly resonate with. So thank you so much on behalf of the community, and definitely we'll keep in touch and hopefully have you some other time again. So best wishes with your program.
Thank you so much, Wealth, and thank you to everyone on this call. I really appreciate your questions and your kind words. And Wealth, to you and your team, keep doing what you're doing, man. I think you guys are doing an amazing thing, and it's exactly what we need, and we need more of it. This is exactly how we build African science and empower it.
Sure, that's true. So we'll keep doing our best, and yeah, we'll stay in touch.
Awesome.
All right, Michael. Yeah.
Yeah, so thank you everyone. With that, I think call it a day or a night or wherever you're in. So thank you everyone. Have a good one. Yeah. Bye.